Restriction means in molecular genetics or in the molecular biology, there are enzymes called the restriction enzymes, right? Already we discussed about this one. Restriction enzymes means some enzymes will cut the DNA in a specific site, right? Those are called the restriction enzymes. What are the restriction enzymes? Echo, R1, okay, BAM, right? HIES, like that. We have different types of the restriction enzymes, restriction endonucleases, right? So these restriction enzymes will cut the DNA in a particular or specific place. That is a restriction endonuclease, a restriction enzyme. So restriction fragment length polymorphism. So here we are cutting a fragment of the DNA, then we will analyze the length. Okay, polymorphism is different types of length we have. Got it? See, for example, it is simple in simple terms, it is just measuring the length of the DNA fragment. That's it. Got it? Just measuring the length of the DNA fragments. Okay. See, for example, let us assume the DNA fragment of some normal gene should be 200 base pairs. Okay. When I'm just for assumption. Okay. A normal gene length should be 200 base pairs. Okay. So when you cut that particular genome, okay, when you cut that particular genome using this restriction fragment, then that should be 200 base pair length, right? But if this restriction enzyme is cutting not at 200 base pair length, it is cutting more than 200 or less than 200, means the gene sequence or the nucleotide number is not correct. So based on the length, we can diagnose the disease. Clear? So what is the RFLP? It's a molecular technique used to detect variations in the DNA sequences, right? Based on the restriction enzyme digestion patterns. Got it? So here, what principle we use? We'll check the length, right? See, if everyone is having 200 base pairs, that is fine. But if somebody is having more than 200 base pairs or less than 200 base pairs means, then we find out the odd one, right? So variations in the DNA fragment length. Okay, so based on that, we will easily find out the disease, right? So here our target is to find out the DNA polymorphism. So what we do in this RFLP, we extract the DNA. Okay, I will show you here what is the procedure of the RFLP. RFLP is a molecular technique that exploits variations in DNA sequences. It involves cutting DNA with restriction enzymes and separating the resulting fragments by gel electrophoresis to identify genetic differences. Just to length, we are measuring the length polymorphism between individuals. Okay, see, for example, a restriction enzyme cuts DNA at specific sequences, right? What are the restriction enzymes we are taking? Echo R1 cuts at GAA TTC. So this Echo R1, E. coli R1, it cuts the gene sequence in this site. If a mutation alters a restriction site, so for example, this is a restriction site, right? But some mutation happened. So when the mutation happens, so what happens? There will be a change in the sequence. Okay. When there is a change in the sequence, then Echo R1, it won't cut in that particular place. So if it won't cut in the particular place means it is cutting some other place. Okay. Then what happens? The sequence of this strand will be bigger or less. The length differs here. Okay. So these length variations can be visualized on a gel and used to detect the genetic differences. Okay. Yeah. See this, you will understand very easily. So this is a normal beta globin. So what is a beta globin? You know, right? So using this RFLP technique, you can diagnose the sickle cell anemia mutation. Okay, whether a person is suffering from sickle cell anemia or whether a sickle cell mutant is there in his cell or in his gene sequence or not. So this easily you can identify. So let us see. Normal DNA sequence contains a restriction site for MST2 enzyme. So what is this MST2 enzyme? So it's an enzyme. It's a restriction enzyme. So this restriction enzyme, this MS2 restriction enzyme can cut this gene sequence. Can you see this gene sequence? So actually this gene sequence is 376 base pairs. 376 base pairs. Okay. But in between, 2001 base pair and 175 base pair, there is a sequence. What is that sequence? That's in that sequence, this MST2 can cut. Okay, so there is a restriction site for this enzyme to cut in this place. Got it? So this is a normal one. This is a normal beta globin gene. Okay, so when you add the MST2 enzyme, it cuts this normal beta globin gene in this place. Okay, if it is cutting, then after that, I mean, when you are doing the gel electrophoresis and later when you map it, or when you see the band, you can find two bands of 201 base pair, 175 base pair, so that it's clear that your enzyme is cutting in that place. So it is a normal gene, right? But mutant globulin. So what the mutant globulin do? The mutant one will change the sequence of this MST2 site. Means here what is there is a base pair, right? So here the base pair mutation will happen. So when the base pair mutation will happen, then the MST2 site, it is unable to cut in this location because it will cut in the specific location only, but that location got muted, right? When the mutant happened, then it is unable to cut there. Got it? So when you find the bigger band, when you find the bigger band on a gel, then you can say that our enzyme is unable to cut in that place. So the band length is bigger. Here we got two small length bands. Here we got a big band. Okay. When you got the big band, here the length matters, right? Here why it is bigger length? Because of sickle cell. Sickle cell. Okay. Then some people may have this heterozygous also. Means I mean, you will be having the bigger band and you will be having the two bands also. Usually we call this as a carrier. Okay. Carrier means their genes contains the heterozygote. So this is expressive. Already, I mean, the people who are having the bigger band are suffering from sickle cell. Okay, so the people who are having the bigger bands as well as the smaller bands means they have both, right? So this is called the carrier. Carrier. Got it? You understood, right? Okay, let us see this. Uh, First of all, you extract the DNA. DNA is isolated from the sample, blood, tissue, or saliva. Then later you cut it with the restriction enzymes, either EcoR1 or BAMH1. Then what this DNA do? DNA will cut in this particular place, right? See, in this particular place, because based on the restriction enzyme, then it will cut in a specific site. Then later you electrophoresis it. The resulting DNA fragments are loaded onto a gel and separated by the size. Okay. Later you transfer it to a nitrocellulose membrane. You know the blotting technique, right? Just like the blotting technique only. So here we are uh, exposing the DNA to an enzyme, restriction enzyme. The restriction enzyme will cut the DNA into different fragments, then separate them based on their molecular weight. You separate them on the gel electrophoresis. Later, you just blot them onto a nitrocellulose paper, okay, or nitrocellulose membrane. Then after that, you add the probes with the radioactivity or fluorescent substances, right? Then you use the autoradiography. Then using this, you can just find the length. Here, what we are doing, it is nothing but a southern blotting, or it is not simply it is nothing but a southern blotting. The same technique, but here after doing the autoradiography, you are just measuring the length of that DNA fragment, right? So here the same thing, right? Sample collection, then DNA extraction, then use the restriction enzyme, then do the gel electrophoresis, and later you bind with the probes which are already having the fluorescent dye. Later and analyze the patterns based on the bands, right? If there is no polymorphism, then nothing will happen. If there is any polymorphism, then you just assay like what type of polymorphism it is having. Okay. So if the sequence is normal, the enzyme cuts shorter fragments, right? If there is a mutation, the enzyme cannot cut. Then if it won't cut, then you can see the larger fragments only. Okay. So where we use this one for genetic testing, for detecting the mutations. Simple thing, RFLP, it's a restriction fragment length polymorphism. Clear? Cut with the enzyme. So if you are getting small bands means that is okay. If you are getting big bands means the mutation happened, then your enzyme is not cutting in that particular space or particular sequence. Clear? That's it. So this is about the RFLP. Okay. So you can expect this as a small question, right? Okay, then let's see the other topic, that is fish. What do you mean by fish? Fluorescent in situ hybridization. Fluorescent in situ hybridization. So what do you mean by fluorescent in situ hybridization? Oh, one second, let me put it again. So fluorescent in situ hybridization means, again try to read the the heading multiple times to understand. Fluorescence in situ. What is the fluorescence? Emission of light by fluorescent dyes, right? The fluorescence means emission of the dyes, emission of the light, right, by fluorescent dyes. In situ. In situ means in the original place, in the same place, right? Means in the cell or in the tissue. In situ. Then hybridization. Already we discussed about the hybridization. What is an hybridization? Binding of a complementary nucleic acid probe to a target DNA or RNA sequence. That's a hybridization, right? I mean, you are adding a probe, right? So what this probe do? The probe will, this is artificial probe you are creating, or you are extracting the probe somewhere, right? The nucleic acid probe. So this nucleic acid probe, you are binding it to a target DNA or RNA sequence. That is hybridization, right? So what is the fluorescence inside hybridization means? Within the cell means here you are not extracting the RNA, you are not extracting the DNA. Okay, the RNA or DNA is present inside the cell only, or inside the tissue only. So there only you are sending the DNA fragments, right? Or DNA probes or nucleic acid probes. Okay, then these will go and hybridize in the same location. Okay, and whenever you are sending with a fluorescent dye, right? Simple. The cell is having some DNA sequence, or let us assume you are suspecting some target DNA is present in the cell or in the tissue, right? Then add a fluorescent dye to the probes. Then these probes will be inserted into the cell. Okay, then what happens? After entering into the cell, these probes, which are complementary to the DNA sequence, they will bind, right? Then later you visualize the genes. Okay, if there is any, I mean, the fluorescence, if it is emitting the fluorescence means it binded. If it is not exhibiting the fluorescence means it's not binded. Okay, okay, let us see what is this fluorescence inside hybridization. Fluorescence inside hybridization is a powerful cytogenetic technique. Okay, it's not molecular biology, cytogenetic. What do you mean by cytogenetic and molecular biology? Molecular biology means it is related to DNA and RNA. Cytogenetics means cell. Cyto means cell. Okay, so here we are sending the probes. Okay, or we are attaching the probes to the genetic material present in the cell. Right, we are not extracting the DNA outside. Right, that's the reason we are calling it a cytogenetic technique. So it's a cytogenetic technique used to detect and localize the specific DNA sequences on chromosomes. Okay, we are not extracting the DNA from the chromosomes, right? In the chromosome, the DNA sequences are there. So you want to detect or localize the specific DNA sequences which are present in the chromosomes. It is widely employed in medical genetics, oncology, and research to identify the chromosomal abnormalities or gene amplifications or deletions or translocations. Okay. Then fish involves hybridizing fluorescently labeled DNA probes to specific target sequences in fixed cells or tissue sections. Means we are not extracting the DNA or RNA, right? So what is the sample required here? We don't need the DNA, RNA, or complementary DNA, or we are not going to extract the DNA. We need the cells or tissues or chromosomes. Okay. Then what we do, we will be adding the fluorescent labeled DNA probes to the specific target sequences in fixed cells. The fluorescent signals indicate the presence or absence or rearrangement of the specific DNA regions, right? Providing insights into the genetic abnormalities. Okay. Let us see the steps in the FISH technique. First, sample preparation. Here we don't need the RNA, right? RNA, DNA. We just need the cells or tissue samples. Maybe blood, bone marrow, amniotic fluid, tumor tissues are collected. Then these cells are fixed onto a glass slide. So how to fix these cells? These cells are fixed onto a glass slide. That is called the fixation. Usually we call this fixation methods. So usually these things we do in the histopathology. I mean, in a cancer hospital. So we used to perform the FISH techniques regularly. every day, like 20, 30 fish cases we used to do. Okay. So, I mean, the fluorescent in situ hybridization, right? So what we do, we'll fix the cells using a methanol acetic acid combination onto the cell. Why fixing? Because if you wash, then the content on that slide may get washed away, right? So to avoid that one, on a glass slide, after adding the tissue, okay, after adding the tissue or cells, usually what we do, we will add just like a thin layer of cells, okay. Then after adding that, then we will be fixing that to the cell. It is fixing is nothing but placing I mean, fixing to the slide. It's like pasting, right? It's like gum like. Okay, so if you peel it off, also it won't come like that. We will be fixing very tightly or hardly the cell component to the slide. So how to fix that one? We use a methanol or acetic acid combination. So that works as a fixing agent, right? Then, and one more thing here, whenever you are collecting these cells, okay, so if the cells are in the interphase, you know about the meiosis divisions and mitotic dividends, right? Interphase, metaphase, right? So we should stop the cell growth. We should stop the cell growth here. Okay, so whatever the cells you are taking, okay, they should be in a non-dividing state, because when the cell is having full, then you can find the complete chromosomal material. When it is a dividing state, sometimes the division may not be complete, so that you cannot find the complete DNA sequence, right? That is the reason the cell should be not in a dividing state. Okay, then if it is a dividing state, then you have to arrest that one. Okay, usually we will be taking non-dividing cells only. Then later you have to denature the sample. Okay, why denaturation? Because the double-stranded DNA, if it is having the double-stranded DNA, then the probes, it won't bind to the DNA, right? So that's why what we do, we will be heating that one, or I mean, you will be heating on that, or else you can use some solutions like formamide. Okay, so use the formamide. Then using this formamide or heating, so what happens here? I mean, you are adding the probes, right? So if you are adding a double stranded probes also, these probes will get denatured into single strands, and the DNA also, it will be denatured into single strands. Okay. So the first one is sample preparation. Later you are denaturing the DNA. Then after denaturing the DNA, then you are adding the probes with the fluorescent dyes. Okay. Then these probes will go and they will attach to a targeting site, maybe a single gene or maybe a centromere, or sometimes, we have different techniques. Okay, it may uh, paint the complete chromosome. Okay, it may, because you are adding the, it's not a small probe, right? It's some thousands of base pair probe you are adding. Okay, so this will go and it will completely complement the entire chromosome also, so that it will paint the entire chromosome. Painting means nothing but, I mean, it hybridizes, right? Hybridizing means already it is having a fluorescent dye, right? So that is the reason it is completely painting a chromosome. Okay, then we use some conditions like changing the temperatures and salt concentration. Okay, and will be incubated for 1 to 1.5, or sometimes it may take 2 to 3 days also, based on the sample. Okay, usually 1 day. Okay, so we do the hybridization, means after incubate for 1 day by providing all the conditions, then the annealing will happen. Annealing means this foreign complementary DNA, it binds to the, or the probe DNA will binds to the actual DNA, right? Then later you wash it. Wash it means unbinded DNA probes will be washed off, right? Then after that, you just detect it, okay, using fluorescent signals, like you use a microscope also. Okay, you just visualize and then you analyze and interpret. Okay, it's a locus specific probe. I mean, we have different probes. So locus specific means for locus, for specific DNA segment, we are using this probe. If it is centromeric probe, then it's links only to the centromere part of the chromosome. Okay, then if it is a whole chromosome paint, means we are using a big base page, then it will paint the complete chromosome. Okay, then you can see the arrangements, rearrangement, translocations also here. Then telomeric probe, it binds to the end part of the, the tip or end part of the chromosome, right? So binds to the end of chromosomes. Like this, we have different types of probes. Okay, let us see an example here. Using this fish, we'll find out the CML. What is the CML? Chronic myeloid leukemia. Usually in children. I mean, when I worked in cancer hospital, so usually we get these samples like CML, chronic myeloid leukemia. So how this chronic myeloid leukemia is diagnosed using the fish technique only. Okay. So what is this CML? CML means it is purely the genetic disorder only. Okay. It's a Philadelphia chromosome. You know this one, right? Philadelphia chromosome. So we use this fish method to detect the translocation in bone marrow. See, actually what happens, uh, chromosome 9 and 22 are there. I think you know about this Philadelphia chromosome, right? So this is a sequence. What is the gene sequence here? Chromosome 9 and chromosome 22. But translocation will happen. Translocation means, see, there are two genes. Okay. BCR gene is on chromosome 22. BCR gene is on chromosome 22. ABL gene is on 9. Okay. But translocation will happen. Translocation will happen. Means, what is the translocation here? These two will get added. Okay. Chromosome 9 and these two will get added. Right? Resulting in the BCR ABL fusion gene. Okay, these two will get added. Actually, BCR should present on the chromosome 22, ABL1 or ABL gene present should be present on 9. But translocation happens. Okay, so you need, when the translocation happens, then the patient will get this CML, chronic myeloid leukemia. Okay, so this is the problem, right? Then you had to find out whether this translocation happened or not. So first you collect the sample, okay, and then you add two probes. Two probes you add. What is the probe? One is the BCR probe, which is labeled with a green color, which are labeled with a green color fluorescent dye. Then you are adding one more ABL gene, that is chromosome, I mean, that connects with the, I mean, that complements with the chromosome, I mean, on chromosome 9, it's a red color. Okay. Simple here, just try to understand. Two genes are there. One is the BCR gene that is present on chromosome 22. Then ABL gene that is present on 9. Okay. Now, you have to check whether really the BCR gene is on chromosome 22 and ABL gene is 9, or they are translocated. Translocated means these two are combined or fusion. Is there any fusion of these two? Okay. So you have to find out that one, whether these are existing alone or independently, or there is any fusion between these two. Okay. So we know the BCR gene sequence, right? Then we'll be taking a probe that will bind only to this BCR gene. Then that probe, you are adding a color called the green color. Then you are taking one more probe, okay, that will be complementary to ABL1 on chromosome 9. Okay, then this is labeled with the red color. So we are adding two probes, one probe with green color, the other probe with the red color. Okay, then you take the sample, denaturate, and add the probes. Okay, then after you see this one, after adding the probes, what happens? You allow some time to complement it, right? So this green color probe will complement the BCR, red color probe also it complements the ABL. Then after that, you wash it. So excess unbinded probes will be washed off. Then after that, you check under a fluorescent microscope. See this? So for example, this is a cell. So usually this cell should have two ABL genes, two BCR genes, right? See, two ABL genes and two BCR genes the cell should have. Okay, so this is a normal cell. Why? Because the fluorescent rays are complementing to two ABL and two BCR, right? So there is no any abnormality in the cell. It's clear. Okay, but when you see this cell, so what is happening? It is showing red color. Means one BCR is sorry, this is an ABL, right? Yeah, one ABL is normal. Okay, then the other BCR also normal, but here there is a fusion. It is showing with a different color, right? So the combination of red and green gives a yellow, right? So it is showing the yellow. Means what is happening here? There is a fusion of the ABL and BCR gene. So it's an abnormal one. So these patients will exhibit the CML, chronic myeloid leukemia. See this? This is also an abnormal. So this is also abnormal because there is a fusion, right? There is a fusion. Okay, this is the fusion. Those are all the abnormal cells, and this is only the normal cell. Okay, so by this you can easily understand the CML. I mean that particular chromosome undergone the fusion or not. Okay, see these are steps. First you start, collect and fix the cells on the tissue. I mean the tissue or cell on the slide, and then denature. Then apply the fluorescent labeled probe. Then hybridization will happen. Okay, within 16 to 24 hours. Then wash off. Then counter stain with the DAP. Sometimes these are already hybridized probes are there, right? Then you have to counter stain to get more fluorescent effect. Then visualize the signals under fluorescent microscope. Then analyze the signal patterns. Interpret the result whether normal or abnormal. Okay, so we use this in the oncology, then prenatal diagnosis also, hematology, genetic disorders like that. So this is about the fish fluorescence in situ hybridization. I hope it's clear. Okay. Now we'll see one more technique, that is microarray. They're all like short answer questions only. So DNA microarray. So what is this DNA microarray? We also call this a DNA chip technology. DNA chip technology. Okay. So actually to analyze the expression of thousands of genes in a single reaction, okay, very quickly and in an efficient manner, we use this DNA microarray technique. Okay, means, see, just now we find out the Philadelphia gene, okay, the fusion of the ABL and this one, right? So that is one. So, but uh, to find out multiple disorders at a time, okay. See, for example, you want to check the what type of chromosomal or what type of gene mutation is present in that particular patient. Okay, maybe the glucose, I mean the lot of gene mutations will happen, right? So to find out all the gene mutations at a time, not a single disease, right? Not a single mutation. You want to find out all the genetic errors in that particular person. Then we are using the DNA microarray technique. Okay. So what is this microarray technique? Microarray technique means, see, we also call the DNA chip technology, to analyze the expression of thousands of genes in a single reaction very quickly and in an efficient manner. Okay, we use this DNA microarray technique to understand the genetic causes for the abnormal functioning of the human body, the entire, functioning. Okay, wherever there is abnormality in the gene, you can find out using this DNA microarray. Right? No need to do multiple tests. Then to understand which genes are active and which genes are inactive in different cell types. Okay, because if some genes are not expressing, means, right, DNA DNA is having the information, right? DNA will carry the information, right? So that information will be given to the mRNA. What the mRNA do? mRNA will carry the information. There's a message, right? It carries the message from the DNA, and based on the message, it will synthesize different types of the proteins required for our body, right? So if the message RNA is not getting a proper message from the DNA, then what happens? This messenger RNA, it won't produce that particular type of the protein. So you will be lacking that protein, right? So if you are lacking some proteins, you will get some genetic disease, right? Or if you are mismatching, means the DNA will be sending a wrong message to the mRNA, or because of some mutation in the DNA, so it is giving some false information or maybe a misinformation to the mRNA. Then this mRNA will give or it synthesizes a false protein, means that protein, it will give a different protein, right? So because of that protein also we'll get some diseases, right? So usually this is the sequence, right? DNA will give the message to the mRNA, then mRNA, based on the message got by the DNA, it will synthesize different types of the protein, right? So this is the way. So if the DNA is not giving the information to the mRNA, or so if, I mean, usually this is called the expression. What is an expression? DNA is giving the information to the mRNA. Then based on the information got by the mRNA only will synthesize different proteins, right? This is called the expression. Okay, the mRNA is expressing different types of the proteins. Clear? So here what we do, we'll take the entire genome. We'll take entire genome and we'll see whether all the DNA fragments expressing the correct, okay, expression or not. Okay, so we'll be testing the entire genome, okay, using this microarray. Got it? So we are testing the thousands of gene expression at a time. Okay, not a single gene sequence. We are testing the entire gene sequences, whether their expression is correct or not. Okay, so here we are doing the entire genome testing, right? Then how to do this one? See, for example, there is a gene A. It's a normal cell, it will produce two proteins. Then gene B, it should not produce any type of the protein. Then gene C will, it has to produce only one protein. So this is a normal, I mean, just it is example. So when you take the cancer cell, gene A, it's not exhibiting, it's not expressing anything. Gene B should not produce any type of the protein, but here it is exhibiting three type of proteins, or it is expressing three type of proteins. Then gene C, it should exhibit only one protein, or it should express only one protein, but here it is expressing two different types of the proteins combinedly. Okay, like this. So the expression will change. That leads to a cancer cell, or that may lead to any other, genetic disease. Okay. So what is a DNA microarray? It's an orderly arrangement of thousands of identified sequence genes printed on an impermeable solid support, usually glass, silicon, or chips or nylon membrane. Okay, so DNA microchips are known as DNA chips or DNA arrays or biochips. Okay, so these are the people behind this DNA. First of all, 1975, Southern blotting, he has developed the blotting technique so that, I mean, you can cut and you can blot, we can separate, right? Then later the DNA microarrays begin in 1980s. Okay, then I think recently in 2002, I think 2022, 2000, now it is 24, right? 26, 2022, I think. Yeah, 2022. 4 years back, a person got Nobel Prize in the DNA microarrays. Okay. So pin-based robotic system also developed in 1990. Then Steve Fodder developed scanner for reading the output. Then quantitative monitoring of gene expression patterns with a complementary DNA microarray reported to Patrick and colleagues in 1995. Then Mark Sheena was proclaimed as the father of microarray technology. Okay, Mark Sheena, he is considered as the father of microarray technology. It's a historical background. Okay. I mean, just I will explain what is this microarray. This is very, very important because once you understand, then you never forget it, right? See, we have a genome. I mean, for example, there is a DNA, right? So, in the DNA, coding parts are there, non-coding parts also there. You know this one, right? Coding areas and non-coding areas. For example, uh, there is a DNA. Okay, in the DNA, 10,000 coding sequences are creating mRNA. So these mRNA will release the protein, or they synthesize the protein. Okay, 10,000 proteins you are synthesizing. Let us assume. Okay, so 10,000 DNA sequences. So you cut the 10,000 DNA sequences, means 10,000 fragment. Each DNA fragment, each DNA fragment will generate one mRNA. That mRNA will release or synthesize a protein. Okay. Now we have 10,000 DNA fragments. Each fragment can express one type of information. Right. Now we have 10,000, and that too we know the names. The first fragment will synthesize this type of protein. Second fragment will, synthesize this type of protein. Third fragment will synthesize, means already we know, right? So we know, right? So these 10,000 fragments, these 10,000 DNA fragments, we are punching, okay, on one sheet, okay? That is called the microarray. See this? So can you see there is one microarray, okay? So we also call this chip, the chips, right? So, I mean, the electronic chips, right? So what we are doing, we are punching thousands of DNA probes onto a chip. Okay. Now we know the expression of first gene. So first gene will express this type of protein. The second gene will express this type of protein. Third gene will express this type of protein. So we already know, okay, known, DNA probes we are fixing on a chip. So this is called a labeled, I mean, this is the generally microarray. Got it? So it contains the entire genome, maybe 10,000 or 20,000, 20,000 spots also you can keep. Okay. So how to prepare this DNA microarray means the solid supports used are glass, silicon, or nylon membranes. This may be a glass or a nylon membrane, or it's a glass also. The length of the slide should be 25 into 75 mm. Right? Now, not even 1 centimeter, right? So it is less than 1 centimeter, where within the area you can keep means 3.6 centimeter square. Okay, if you take that one, it will be 3.6 centimeter square. Then 10,000 to 20,000 spots, genes we can place. Okay, means DNA fragments we can keep. Then diameter of each spot is 50 to 150 micrometers. Okay, so what is the diameter of each one? 15 to 50 to 150 micrometers. Then distance between the spots is 200 to 250. Means we should follow a sequence, right? So the distance should be this much, and the DNA, the spot should be this much. So we have some specifications. So based on that, you are creating one microarray slide. Okay. Then how to punch? How to add this? The DNA fragments here. Means we have different techniques. One is photolithography. So using this photolithography, we are punching, like this. So let us assume this is a plate. In the plate, this is a DNA sequence. So you are adding the DNA sequence like this. Okay. These are all the DNA sequences, known DNA sequences we are adding. There is a robot spotting also. Okay. Using the robot spotting, it will punch or it will place in that particular microarray. Or there is a inkjet spotting technique also there. Okay. So it will keep like this. Okay. It will keep the nucleotide sequences like this. Okay. Like this. Clear? Now, uh, how to find out, how to use this one? I hope you are getting right. What we do, we create a microchip. What the microchip contains? Microchip contains 10,000 to 20,000 known DNA fragments, right? So 10,000 to 20,000 DNA known fragments means those are all the coding only. We know like which one will release or which one will express what type of protein. We know already this one, right? So later what we do, for example, we are taking the sample from a normal person and the diseased person. Okay, we are taking the sample from a normal person and the diseased person. Then you extract the DNA, extract the RNA. Why RNA? Why not DNA? See, DNA will give information to the mRNA. mRNA will carry the information, right? So based on the information present in the RNA only, it will it will express the type of protein, right? So DNA, it won't directly express the protein, right? So mRNA will be expressing the protein or expressing whatever the information it is carrying from, right? So to know whether this mRNA is really expressing the protein or mRNA is keeping silent. Okay, though it get the information from the DNA, so the mRNA is kept silent. It's not releasing any type of the protein, right? So that is the reason we are not finding the DNA sequence. So we are using the mRNAs because mRNAs only they have to express, right? So that is the reason what we are doing. We are extracting the RNA. If not, if you are extracting the DNA, then build a reverse transcriptase. I mean, usually extract the RNA, mRNA. Okay. Then after extracting the mRNA, then you have to build one complementary strand. When you build a complementary strand on the RNA, what happens? That is nothing but the complementary DNA, right? Then add the color. So green color you are adding to the normal tissue, then red color to the abnormal tissue. Okay, this is a tissue. Then keep these two or hybridize these two on a microarray chip. Okay. Then what happens? What happens in the microarray? See, when this person, when uh, there is a green color label means when there is a green color label means the normal tissue probe and the normal tissue probe got hybridized with the known one. Okay. If you add a diseased tissue, then if it is having green color means the diseased tissue is hybridized with that particular one. Got it? See, here let's take an example. Two samples are there. Centrifuging and then supernatant after extraction, then you get the RNA. Okay. Remove the buffer that contains the RNA, then you do the reverse transcriptase, it gives the cDNA. Okay, complementary DNA. Then add it onto the microarray. Okay, microarray slide. After adding, because they are having the probes, right? So we're adding the fluorescent probes. So when green color comes, so it indicates that the normal person probe got added here. Okay. Then when the red color comes, then the abnormal patient, I mean the patient, that particular one added here. Okay. Because what we do here, see, in this, in this standard microscope side, we keep the normal and abnormal also. You got it? Means here we keep the normal genes also and abnormal genes also will keep. Clear? It contains normal gene expression and abnormal gene expression also. Okay. So if normal gene, if it shows green color means that normal gene got complemented. If the red color comes means the abnormal gene is complemented. Okay. So merged means if you have both, then we can say that this is having normal as well as abnormal also. Okay. Then here we will measure based on the intensity, fluorescent intensity. Okay. So we use this in the gene expression profiling, toxic studies, then induce pluripotent stem cell lines. Okay. So this is the microarray. You understood, right? Simple. This microarray is a high throughput technique. Okay, use it to analyze the expression of thousands of genes simultaneously, right? So here what we do, what is the principle? Thousands of DNA probes fixed on a chip. Later we'll hybridize with labeled DNA or complementary DNA samples. Here our target is entire genome we are testing. Okay, so what is the sample required here? We need RNA or complementary DNA. Okay, then what is the method here we use? Laser scanner or computer analysis, right? Then thousands of genes simultaneously we can find out like their expression. How long it will take? Maybe few hours to 1 day it will take, right? But the cost is more because we are using thousands of 10,000, 20,000 of probes, right? No DNA sequences in a microchip. So this is called complete gene expression profiling. Okay, if the man is having any abnormality, you can find out.